This is a revision video on aerobic respiration. Pause the video and answer the 7 revision questions, and when you're ready you can start the video again to go through answers. Okay, so question 1. So you should have written out the main steps of glycolysis and then stated where glycolysis occurs. So for the process of glycolysis you should have had this. Remember, that process of adding a phosphate group to a molecule is called phosphorylation. So remember, for each trisphosphate molecule during that process of producing pyruvate, 2 ATP produced for each of those molecules. So what you should recognize from that is that from the process of glycolysis you get a net production of 2 ATP molecules, because 2 ATP were used at the start for the phosphorylation of glucose. And when does this process occur? Cytoplasm. Okay, so let's move on to the link reaction. So the link reaction occurs in the mitochondrial matrix. So the steps for the link reaction you should have are initially really important to remember that pyruvate is produced from glycolysis. That's in the cytoplasm. It needs to be transported into the matrix, and that occurs via active transport. So again, really important that you remember that removal of carbon dioxide is a decarboxylation process. Pyruvate is then oxidized, and that produces okay, so that's the link reaction. Let's move on to the Krebs cycle. So we start with acetyl coenzyme A that was produced from the link reaction, and so let's go through the steps of the Krebs cycle. So the first thing is the coenzyme A is removed, and that can return to the link reaction to be used there. And that combines with oxaloacetate to form a 6-carbon molecule. Okay, so we've got a decarboxylation and oxidation of that 6-carbon molecule to produce a 5-carbon molecule, and then the next step and so a lot goes on between the conversion of the 5-carbon molecule back to oxaloacetate, that 4-carbon molecule. So there are 2 reduced NAD that are produced from the oxidation of that molecule, and there's the production of 1 reduced FAD, another oxidation reaction of the 5-carbon molecule. And then we also have ATP produced, and that is called substrate level phosphorylation in this case, to differentiate it from oxidative phosphorylation, which we'll talk about in a moment, and there's also another decarboxylation reaction. So that's the Krebs cycle, and obviously leads to the regeneration of that oxaloacetate that can then be reused again to form that 6-carbon molecule, and the cycle can continue. It's also known as the citric acid cycle and the tricarboxylic acid cycle as well. Where does that occur? In the mitochondrial matrix. So now let's move on to the electron transport chain and oxidative phosphorylation. So firstly, where's the electron transport chain found? It's found embedded in the inner membrane, or cristae. And so let's go through the process of oxidative phosphorylation. So we've got the electron transport chain embedded in the membrane and ATP synthase. So we start with reduced NAD, and there are several sources for that reduced NAD, and I've given you the values of reduced NAD produced from glycolysis, the link reaction, and the Krebs cycle. And so the important thing that happens to that reduced NAD is that it gives over the hydrogen atoms that it carries, and they split into an electron and a proton. We also have reduced FAD, 2 of those from the Krebs cycle per glucose molecule. And again, that gives over its hydrogen atom, that is an electron and a proton. So let's go through the next step. And with these next steps, I'll provide text to describe some of the key events that you should have written down. Okay, so they were the key steps of oxidative phosphorylation. So now we'll move on to working out how many molecules of ATP are produced from this process. So for question 5, per reduced NAD, you get 2.5 ATP, and per reduced FAD gives 1.5 ATP. So now let's go through the amount of ATP produced using the table. So we'll start with the glycolysis row. So during glycolysis we get 2 reduced NAD, no reduced FAD, and 2 ATP. And we know that those reduced NAD equate to 2.5 ATP. So that's 5 ATP and then 2 ATP produced from substrate level phosphorylation. So in total we get 7. The link reaction gives us just 2 reduced NAD per glucose molecule. So that equates to 5 ATP. And from the Krebs cycle, and so in total with those numbers we get 20 ATP from the Krebs cycle. So we can total those up for each. And if we do the maths for that, from a single glucose molecule during aerobic respiration, 32 molecules of ATP are produced. So let's move on to the answer for question 7. So in the middle there, I've provided a complete diagram of glycolysis, the link reaction, and the Krebs cycle. And what we're going to do is use that with our explanation. So if we start with proteins, they are hydrolyzed to form amino acids. Those amino acids go through a process called, so that NH2 group is removed and it can be excreted as urea. And so the remaining carbon skeleton can form either, and that's dependent on the R group, obviously. And then those compounds can be converted into molecules that can feed into respiration at various points. So 3 carbon compounds can convert it to pyruvate, 4 carbon compounds oxaloacetate and can go in at the Krebs cycle, and the 5 carbon compounds again can form the 5 carbon compound found in the Krebs cycle. So for triglycerides, you should know that they can be hydrolyzed to glycerol and fatty acids. So with glycerol, it's phosphorylated, and you should remember that glycerol is a 3 carbon compound. And so this phosphorylated 3 carbon compound can be converted to triose phosphate. And so that's how glycerol can feed into aerobic respiration. And finally, fatty acids. So here's an example of a fatty acid, this long hydrocarbon chain. And what happens to that is that, and so as you can see, we've got these 2 carbon molecules that can be extracted from a fatty acid, and each of those can be converted into acetyl coenzyme A and can be used as a respiratory substrate there. So that's it for aerobic respiration. As always, read the textbook pages that are in the description to help you fully understand this, and go through this as many times as you need in order to be getting this correct and be ready to do questions in the textbook and, of course, exam questions.